गुड आफ्टरनून बच्चों, कल आपको बता रहे थे पीसीआर के बारे में, उसी को आगे कंटिन्यू करते हैं। पहले बात करेंगे पीसीआर इक्विपमेंट की। द पीसीआर इक्विपमेंट कंसिस्ट ऑफ अ थर्मल साइक्लर टू प्रोग्राम द टेम्परेचर एंड टाइम पैरामीटर्स फॉर ईच ऑफ द 3 स्टेप्स ऑफ पीसीआर, डीनेचुरेशन, अनीलिंग और एक्सटेंशन जो होती है। The thermocycler contains a heating block with necessary software for the investigator to set the appropriate temperature and the time interval. The heating block holds the microcentrifuge tubes that contain the PCR mixture. The heating block is designed to ensure consistent temperature across the entire volume of the reaction mixture, both with the microcentrifuge tube and between multiple microcentrifuge tubes. Now comes the determination of length of the copy DNA, that is the cDNA or the complementary DNA. An allocate of the cDNA is analyzed by agarose gel electrophoresis to determine the BP length, and the BP length is equal to the number of the nucleotides in the cDNA product. Commercially prepared DNA size measures run in the parallel with the PCR allocate. The electrophoretically separated pattern is stained with the fluorescent dye ethidium bromide. The fluorescence can be photographed by using a Polaroid or a digital camera. The BP length of the PCR product can be measured by comparing the PCR signal on the gel so that on the closest of the DNA size marker with its co-migrates in electrophoresis. A successful PCR is realized when only one cDNA signal represented by a single band on an agarose gel is observed at the predicted BP length. This signifies high specificity between the primers and the target DNA sequence. If the primers anneal erroneously to the regions of DNA other than the target sequence, a PCR cDNA product will have an incorrect nucleotide sequence. Therefore, observation of the predicted size of a PCR product by electrophoresis is necessary. DNA sequencing should be performed to document the performance of the correct nucleotide sequence of the PCR product and calibrate its BP size determined by electrophoresis. The PCR inhibition includes the effect of the heat denaturation on the extracted DNA, the DNA extraction methods which may inhibit DNA polymerase. These may cause low yield of the cDNA and may be observed in separated electrophoretic patterns. False positive reactions in RT-PCR may also be observed, sometimes by the generated presence of genomic DNA, that is, the genomic DNA contains an intron and exon sequences while the cDNA contains the exon sequences only. The treatment of the extract with RNA as removes the genomic DNA. The practical applications of PCR are for the amplification of the DNA, for identification of a target sequence, synthesis of a labeled antisense probe, identification of DNA or mRNA, that is messenger RNA, present in low concentration on within the viable cells in culture in situ PCR. In situ PCR is a potentially powerful tool in molecular pathology because gene mutations associated with cancer can be identified with a tissue biopsy before the histological observation of the dysplasia or malignancy. The PCR cDNA may also be converted to a probe to detect DNA or its mRNA transcripts using southern and northern blotting techniques. Now next process is gene cloning. Gene cloning is performed by using restriction enzymes. Without these enzymes, molecular biological analysis involving gene cloning would not have been possible. These enzymes are widespread in bacteria in which they fulfill adhesive function, cleaving the molecules of the foreign DNA. The usefulness of these enzymes is derived from the fact that they do not cleave the cDNA at random but recognize and cut specific nucleotide sequences only. Some enzymes cut the cDNA or the tDNA in such a way that blunt ends are produced, while others, for example E. coli RI, cut the asymmetrically so that sticky ends are left, which are extremely useful for re-annealing the fragments to produce recombinant cDNAs. The gene cloning methodology involves the DNA molecule under the test is fragmented by using enzyme restriction endonucleases. The single-stranded fragments are separated on a polyacrylamide gel by electrophoresis. The sequencing of DNA is performed by using chain termination method of F. Sanger or the chemical cleavage method of A. Maxim or Gilbert. The separated pattern of DNA on a polyacrylamide gel can be transferred to a piece of nitrocellulose paper or the southern blot technique. This paper can be treated with a radioactive DNA molecule acting as a probe to discover the separated pattern. After confirming the base sequence, the DNA fragment of interest is introduced in a plasmid by cutting it by using restriction endonucleases. The DNA fragments with the complementary sticky ends of the plasmid are annealed by using the enzyme DNA ligase. This recombinant plasmid is then inserted in the bacteria such as E. coli and allowed to replicate under appropriate growth conditions in a specific media. The replicated clones of the DNA within the bacteria phase are subsequently recovered by harvesting and lysing the cells. Frequently, an attempt is made to isolate the mRNA transcribed from a desired gene. For example, proinsulin mRNA can be isolated from pancreatic beta cells. It is attached to the polysomes. The polysomes are precipitated by using their specific antibodies. Separated mRNA can function by using the enzyme restriction endonucleases, and the base sequences in the fractions can be identified by southern blot techniques. When a fraction containing the desired mRNA has been identified, it is used to direct the synthesis of the DNA molecules or the cDNA complementary to all of the mRNAs, that is the messenger RNAs in that fraction. This cDNA can be used as a probe which can direct synthesis of insulin. The enzyme reverse transcriptase can synthesize a DNA strand complementary to a mRNA template by using a mixture of 4 deoxyribonucleotide triphosphates, that is DATPs, DTTP, DGTP and DCTP, which provide a short length of primer, that is a short length of the DNA is base paired with 3 ends of the RNA. Since mRNA has a poly tail at its third end and a short oligo molecule will act as a primer for reverse transcriptase. This single-stranded DNA can be used to direct the synthesis of complementary DNA strand. The second strand requires a primer base paired with the poly tail, and enzyme reverse transcriptase is also needed. The production of large quantities of genomic or the cDNA probes is carried out by cloning. If the sequence of the DNA being investigated is known, that is the oligonucleotide probes can be made in order. if they are of an appropriate length of about 19 bases and conditions are carefully controlled. For hybridization, synthetic oligonucleotide probes can be sufficiently specific to recognize a single base change. Another use of synthetic oligonucleotide is as primers in the PCR reaction. PCR has outstepped many of the more tedious techniques of molecular biology. It is essentially a means of cloning DNA without the need of vector or bacteria. Then coming on to the next, that is probe labeling. Radioactive labeling of cDNA is carried out by neck transplantation, in which low concentrations of DNAs are used to make occasional single-strand nicks in the DNA. DNA polymerase then fills in the nicks by using an appropriate deoxynucleotide triphosphate, at the same time making a new nick at the third side of the previous one. The most frequently used radiolabel is phosphorus 32, but iodine I-125, sulfur 35, carbon 14 and tritium 3 have also been used. The method is used in enzymatic incorporation of the label. Single-stranded nicks at randomly introduced each strand by DNAs or endonucleases. Labeled triphosphate nucleotide are then introduced by using DNA polymerase at generated third group in the fifth or the third direction by using appropriate strand at the template. The very high specific activity probes have a short life limited by isotopic decay and dialysis of the probe. Non-radioactive methods of probe labeling are clearly advantageous for clinical applications because they make the probe as a stable reagent. The laboratory personnel are also not exposed to radioactivity, and cost of radioactive waste disposal is also eliminated. Avidin is a glycoprotein present in egg white. It binds with extremely high avidity to small molecules of biotin, and biotin is easily linked to these cross-linkages, protein and nucleotides, to make their further copies. Now this finds its applications in diagnostic microbiology and diagnostic pathology level also. Traditional microbiological methods employ staining techniques, direct visualization, culturing and isolation of the microorganisms, or detection of specific antibodies. However, many organisms are not easily identified by staining. The culture technique is laborious and requires long incubation periods. Some microorganisms require expensive media for culture, and some cannot be cultured at all. Measuring the host antibody response may not be informative during the active phase of the infection. It may not distinguish between the past and the present infection. The nucleic acid probes directly detect the genetic information of the pathogens in the specimen. Nucleic acid is a relatively stable component, hence specimen visibility and the transport time may no longer be important as an issue. With sufficiently sensitive assays, the viral infections can be detected, and probes can also be designed to directly detect antibiotic resistance genes. Infections caused by bacteria such as Mycobacterium tuberculosis, Neisseria, gonorrhea, and infection caused by viruses such as herpes simplex type 1 and type 2, papillomavirus, adenovirus, Epstein-Barr disease, hepatitis B virus, and AIDS virus can be detected by using the appropriate DNA or the RNA probes. अब हम बात करेंगे अगली टेक्निक की, जो कि बहुत ही कॉमन होती है, इसमें आता है कैलोरीमेट्री एंड फोटोमेट्री. The body fluids such as blood, serum, plasma, CSF and urine contain organic and inorganic substances. For example, blood contains organic substances such as glucose, urea, uric acid, creatinine, proteins, etc. It also contains inorganic cations such as sodium, potassium, calcium, and anions such as inorganic phosphorus, chlorides, bicarbonates, etc. Most chemical substances in the blood are in equilibrium if their rates of production and the loss by degradation or excretion are equal. The normal concentrations of most chemical substances vary within normal limits or the normal ranges. The disease may change these equilibria in several ways. The factors which influence the chemical composition of the blood in disease may be broadly classified as physical and metabolic. The physical factors include those cases of retention due to alteration or destruction of the tissues or organs such as kidneys, liver, lungs, and the heart. In kidney diseases, accumulation of nitrogenous substances such as urea, creatinine, etc. in the blood may take place. In a liver disease, bilirubin concentrations of blood may increase with increase in liver enzymes such as SGPT and SGOT. In the metabolic diseases, alteration in the chemistry of the blood may be induced by increased or diminished formation or utilization of various constituents. Thus, in diabetes mellitus, due to the deficiency of insulin, accumulation of glucose in the blood takes place. The detection of biochemical abnormalities in the tissue samples is impracticable. It is easier to access chemically the secondary disturbances in the blood, urine, CSF, and other body fluids. Owing to its number of factors such as small concentration of many chemical substances and the difficulty of isolating them, calorimetric methods are frequently used. In calorimetric determinations, a specific reagent or a specific reagents are used which react with the specific component to form a colored complex. The concentration of the colored complex is directly proportional to the concentration of the component in the specimen. The depth of the colored complex is measured on a photometer or a spectrophotometer. The photometric readings are compared with the known primary standard or with a quality control serum sample. The photometry involves the measurement of the light transmitting power of a solution in order to determine the concentration of light absorbing material present. In order to get introduced to photometry, it is necessary to get familiar with the following terms and components used in a colorimetry and photometric devices. The analytical procedures based upon the direct measure of color intensity in terms of light absorption at specific wavelengths are known as photometric procedures, and the instrument used is called as a photometer. Calorimetric procedures are limited to the visible portion of the spectrum. The photometric procedures, however, involve use of ultraviolet, visible and infrared portion of the spectrum. Sunlight is a type of radiant energy comprising of ultraviolet light, that is below 380 nanometers, of visible light, that is 380 to 760 nanometers, and infrared light above 760 nanometers. As you know, light travels in waves. The quantity of the light is judged by the wavelength of which is the distance between the crests of the waves. It is expressed in the terms of nanometers. 1 nanometer is equal to 10 to the power minus 9 meters. The molar extension coefficient is the extension or the OD or absorbance given by a substance concentration of 1 mole per liter in a light path of 1 centimeter. A monochromator is any system or component that will transmit monochromatic light, that is light of a single wavelength. In case of photometer, a filter is used as a monochromator which may not transmit the monochromatic light but transmits a narrow band, that is 4 to 20 nanometers of the spectral region. However, in the case of a spectrophotometer, a prism or diffraction with a slit is used as monochromator to transmit single wavelength. Light source, a bulb which emits radiant energy for the photometric determinations. A tungsten lamp emits a continuous spectrum of light in the visible range of 400 to 760 nanometers. A halogen or deuterium discharge lamp provides wavelength range of 200 to 900 nanometers, which covers the part of the ultraviolet range, complete visible range, and part of the infrared range. Now we have a cuvette or a cell. It is a round or square tube made up of glass, quartz, silica, or special type of plastic. It holds the solution meant for the determination of absorbance. The width of the cuvette represents the path of the light, which should be preferably 1 centimeters. Photocells are there as photodetectors made up of light sensitive material such as selenium or cuprous oxide. These generate an electrical current which, when light is transmitted by a solution, falls on them. The electrical current is measured by a galvanometer. A colored solution, that is, a solution has color because it absorbs a lesser proportion of the color represented by it. For example, a blue solution appears blue because white light passes through it and the large proportion of the blue light will be transmitted and other parts of the white light, that is green, yellow, red, etc. are absorbed by the blue solution. When yellow and the blue colors are transmitted, the solution will appear green. The solutions for ultraviolet determinations are colorless but contain certain substances such as NAD, NADH, etc. and these substances absorb the UV light in variable proportions. The transmitted light gives measure of the light absorbing material present in the solution. A blank is also there. Absorbance as measured in photometers involves not only the absorbance of the solute in the solution being evaluated but also of all the molecules. A reagent blank contains only one reagent. A sample may be added into it without performing a reaction so that a colored complex will not form. However, if a sample is colored, then a separate sample blank is required, which is prepared by adding the sample to saline or the reagent by excluding a reacting component. The blank solution is used to set the meter of the instrument to 100% transmittance or zero absorbance. Now the basic components of a photometer are a light source, a lens or a condenser, a filter, a cuvette, and a galvanometer or the readout device. A spectrophotometer contains all these components except a filter. Instead of that, it contains a prism or diffraction grating with a slit. The basic steps involved in a photometric procedure are as follows. Firstly, pipetting of the reagent in 3 tubes labeled as test, standard and blank. Secondly, addition of the specimen and standard in the respective tubes and distilled water in the blank. Keeping the tubes for incubation at specified temperature or room temperature about 37 degrees Celsius or 100 degrees Celsius. Reading out the intensities of the test and standard against blank at the specific wavelength. Calculating the unknown concentration or the specimen component by using the Beer's law. The basic steps involved in the working of a photometer are: light transmitted by a light source is focused on a filter through a condenser lens. Filter transmits a narrow band of light. When the narrow band of light passes through a colored solution, the light absorbed is directly proportional to the concentration of the colored solution according to the Beer's law. The light transmitted falls on the photocells, which converts this radiant energy into electrical energy. The galvanometric readout device records the readings in terms of percent transmittance or optical density or OD. Now coming on to the principle on which these photometers work. The working of the photometers and spectrophotometers is based on the Beer's and the Lambert's law. The Beer law states that the optical density of a solution is directly proportional to the concentration of the solution. The Lambert's law states that the optical density of a colored solution is directly proportional to the path of the light, that is the diameter of the cuvette. If the diameter of the cuvette is doubled, the OD will also be doubled. But since same cuvette or cuvettes of the same diameter, preferably 1 centimeter, are used for the photometric readings, automatically the Lambert's law is observed. According to the Beer's and the Lambert's law, the absorbance of a solution containing light absorbing material depends on the following factors. Firstly, the nature of the substance. Second, the wavelength of the light. Third, path of the light. And fourth, the amount of the colored material in the light path. If incident light is denoted by P0 and the transmitted light emerging from the cuvette containing the solution is P, then the ratio of P divided by P0 represents the transmittance T of the colored solution. If the material does not absorb at all, then P and P0 are the same, that is P divided by P0 is equal to T and the percent transmittance is equal to 100. By using the negative logarithm of the T, the measurements are transformed to the light absorbed 1 divided by the transmittance T. The equation now becomes A is equal to minus log T, and since 2 is the log of the 100, then percent transmittance, that is the percent T formula, becomes A is equal to 2 minus log T. In an analytical procedure, when the test is compared with the standard, the ODT is directly proportional to the CT, that is the standard. This formula is used to determine the concentrations of a specific component analyte in all analytical methods involving photometry, where CT is equal to the concentration of the component or the analyte and the sample which is unknown. The instrument which uses a light filter for the wavelength at which photometric measurements are made is called a photometer. In the case of a spectrophotometer, a prism or diffraction grating with a slit is used to get monochromatic light. Filter photometers are less expensive than the spectrophotometers because they require less technical skills in operation and are satisfactory for routine analytical procedures only in the visible region of the spectrum. The light filters do not transmit monochromatic light, but they transmit narrow range of spectral region, that is 8 to 30 nanometers. The spectrophotometers use monochromatic lights. These instruments are more expensive than the filter photometers, and the cost increases with increased sensitivity, spectral range, and narrowness of the spectral region isolated. The spectrophotometers have the advantage over filter photometers of greater convenience and flexibility in choice of wavelength, not only in the visible part of the light but also in the ultraviolet and the infrared regions of the light. Now coming on to the choice of the photometers, इनको हमको select कैसे करना है? For routine analytical purposes based upon established procedures, any good type of filter photometer can be satisfactory. A filter photometer differs considerably in the width of spectral band transmitted by the filters supplied with the instrument. Hence, the photometer which is equipped with the filters and which transmits narrow band of the spectrum is more satisfactory because agreement with the Beer's law cannot be expected with filters transmitting a wide range of the wavelengths. The specifications preferred for the selection of a photometer are that total number of filters, peak transmittance of each filter or the pass band, the cuvette type, whether optically matched glass cuvette or ordinary glass cuvette, and whether they are round or square type. The total number of cuvettes supplied and the minimum quantity of solution required for photometric readings. The cost per test can be reduced by using minimum quantity of reagent. A satisfactory photometer is the one which offers cuvette size suitable for the reading of 1 to 3 ml of the reaction mixture. The number of photocells, that is the photodetector system of single cell photometer contains 1 photocell, and that of a double cell photometer contains 2 photocells. Now the advantages of a single cell photometer is that the manual operations are limited. It is very easy to operate. For the photometric readings of unstable colored complexes due to the fast fading of the color, the single cell photometer can be useful. Up coming on to the disadvantages. The disadvantages of a single cell photometer is that there are fluctuations in the main electric supply. The readings may vary according to the electrical fluctuations. Even if the light source is not stable, there is a possibility of errors due to change from the initial light intensity during a measurement. The advantages of a double cell photometer is that if there are fluctuations in the main electric supply or due to unstable light source, they are nullified due to the 2 photocells in a balanced circuit. The double cell photometers are null point instruments. 2 closely matched selenium cells are balanced against each other so that no current passes through the galvanometer when the actual reading is being made. The fluctuations in the mains supply affects each side equally. The optical density readings, that is the OD readings, can be recorded directly on the scale. The main disadvantage of this double cell photometer is that the manual operations are somewhat elaborate compared to a single cell photometer. The main important components of a photometer or any spectrophotometer are a light source. The function of a light source is to provide incident light of sufficient intensity for the photometric measurements. A tungsten lamp is used in the case of filter photometers where a visible range of light is required. It emits a continuous spectrum of light in the visible range of 400 to 760 nanometers. For work in the ultraviolet region, however, ordinary tungsten lamp is not useful. For such purposes, a high pressure hydrogen or deuterium discharge lamp, halogen lamp, a radioactive radiant range obtained from 20 to 900 nanometers, and a radiant range obtained from 340 to 760 nanometers in the case of a tungsten lamp may be used. Second component is a monochromator. The main important characteristics of a monochromator are its band pass width, nominal wavelength, and the peak transmittance. The usual filters used in the case of photometer are made up of colored glass or gelatin containing suitable dyes. These offer relatively wide bandwidths and low peak emissions. Narrower bandwidths and low peak emissions are obtained with the interference filters. Between semi-transparent silver films, a transparent dielectric spacer or low refractive index is placed. Cutoff filters are placed on each side of these films to control the incident light. These filters have a bandwidth of 8 to 17 nanometers and the peak transmittance of 40% to 60%. Multi-layer interference filters consist of successive layers of high and low refractive index dielectrics on alternating layers. These filters allow band pass width of 4 to 8 nanometers and a peak transmittance of 60% to 90%. The spectrophotometers use prisms or diffraction gratings as monochromators. These devices separate the various wavelengths of radiant energy as produced by a tungsten lamp by refraction or diffraction and from the spectrum produced during the desired wavelength may be selected by the adjustment of an exit slit. The actual separation between the two wavelengths depends upon the dispersive power of the prism. Dispersion by glass prism is about 3 times that of quartz. But quartz or silica prisms are mandatory for ultraviolet determinations. In the case of prisms, the longer wavelengths are not refracted wide and are obtained in the crowded form. The diffraction gratings, however, produce a linear and non-curved spectrum. Then comes the cuvettes or the sample cells. One of the most of the filter photometers, round type cylindrical cuvettes are used. These cuvettes are fairly accurate if they are marked in front of proper light path orientation. Otherwise, these are prone to variable reflection and refraction errors as well as the lens effects. In the case of some spectrophotometers, however, square or rectangular absorption cuvettes are used. These cuvettes have plane parallel faces and a light path of constant length, that is 1 centimeter near about. They are free of optical aberrations. For ultraviolet determinations, these thermocuvettes maintain constant temperature 25 degrees, 30 degrees or 37 degrees Celsius by Peltier effect or thermoelectrically heating or by hot water circulation around the cuvette. In the case of some of the special types of photometers and spectrophotometers, a flow cell assembly with electronically controlled temperatures using the Peltier effect is used. Because the same cuvette is in the light path at all times, cuvette errors would be compensated. A source of vacuum is used to empty the flow cell. In colorimeters and spectrophotometers, simple lenses are used to focus for colliminate the light beams. In special types of spectrophotometers, front surfaced mirrors are used to reduce the energy loss. The surfaces of these mirrors are coated with thin film of magnesium fluoride to reduce the light scattering.